Hello, I'm John Greeley, and I'd like to tell you a little bit about the study we're currently doing on the aging of cells, in particular the aging of our T cells that we have in our blood. So this is a study which is run out of the Einstein Center for Epigenomics, and it is funded through the National Institute of Aging and the National Institutes of Health. So what are we doing? Well, we have a team here in the lab. We're very interested in asking the question, what is happening when cells age, in particular related to what is called their epigenetic properties. So one of the definitions that people have for epigenetics is molecular changes that have to do with the regulation of genes, in particular a type of chemical modification to the DNA called DNA methylation. Now, it's been known for some time that with age there are some places in the genome that will lose DNA methylation and other places in the genome that will gain DNA methylation. And this has been found very consistently, not only across different individuals in the population, but also in different cell types and in different species. But we don't know why it happens. So one of the possibilities is that individual cells actually change their methylation. This is probably the way that you would naturally think of why DNA methylation would change, that the cell used to have methyl groups stuck to the DNA, and it no longer has them anymore. But there are some other possibilities, and a big one that we like to think about is whether the actual cells have changed with age, so that what used to be a large population of cells that had a lot of methylation at that locus is now being replaced by a new subpopulation of cells where there's less methylation at that particular location in the genome, which will give rise to the appearance of a change of DNA methylation, but it will not necessarily be the same as in the last slide, where we saw the individual cell changing its DNA methylation pattern. And this is important to understand as a distinction. So one question that arises is, why is it that specific places in the genome have these changes of DNA methylation with age? It's not something which happens throughout the genome. It's a very specific set of individual locations. This is indeed a conundrum, and one of the models that we need to consider is the possibility that something that acts to bind to very specific sequences in the genome is mediating this. And there's now very good evidence that proteins called transcription factors, which bind to specific sequences, can cause the region locally to lose DNA methylation. But if the transcription factor no longer binds, then methylation is reacquired at those locations. So if you can imagine a transcription factor binding in a younger individual at a particular place and no longer binding later in life, that could be an explanation for why you would gain methylation at that locus. You could also consider the possibility that transcription factor only starts to bind to the locus later in life as you age, and that's why you lose DNA methylation at that site with age. Do we have any idea what transcription factors might be involved? Well, there's one very interesting clue that was from a study published in 2015, where they looked at the locations where the glucocorticoid receptor binds in the human genome and showed that it was enriched at these sites where DNA methylation appeared to be changing consistently with age. So the glucocorticoid receptor is a type of transcription factor which binds to glucocorticoids, and glucocorticoids are produced as part of the stress response in humans. So the model that you would consider is that chronic stress hormone exposure to cells is what causes them to age, which is a very intriguing theory. So how are we going to tackle this? We're going to use a lot of very sophisticated genomic techniques. Whole genome bisulfite sequencing is what allows us to study DNA methylation in great detail. And instead of looking at just a small number of tens or hundreds of thousands of locations or cytosines in the genome, this allows us to look at every cytosine, all 28 million of them. So the bars that are shown there show the relative scale of how much information we'll be getting through our approach. We'll also do regular sequencing, but low coverage of the genome, which will allow us to confirm people's ancestry. It'll allow us to discover the DNA sequences that may be causing people to differ in terms of their DNA methylation from person to person. And we're also developing a new technique that will allow us to measure the lengths of the telomeres in each person. These are the caps of the chromosomes, and it's known that these shorten with age. So it's going to be a nice extra piece of information that we can harvest from the same study. We're going to look and see where the transcription factors are binding in the genome through chromatin mapping studies, and we'll also look specifically at where the glucocorticoid receptor binds in the T cells that we are going to be studying from people's blood. To try to understand whether there are changes in the subtypes of cells that are present in the individual as they age, we'll look and see whether there are changes in the clonality of the cells. In other words, are there specific cells, as shown in red here on the bottom left, where some clones start to take off and become a major component of the population of cells. We'll also do what's called single-cell transcriptomics, or looking at the gene expression pattern of each individual cell to see whether this allows us to define cell subtypes and whether these proportions of cell subtypes change with age. So, why are we studying Ashkenazi Jewish people in particular? Well, it's because of a legacy of a great research done here at Albert Einstein College of Medicine in the Bronx in New York, studying centenarians who, for some reason, were gifted with this incredibly long, healthy life, and prompted studies to see whether there was something about their genetics that gave them this advantage. And this is work performed in Einstein's Institute for Aging Research, led by Dr. Nir Barzilai. In order to study younger individuals, 20 to 40 years of age, we need to collect samples from this group, and they should also be genetically of the same ancestry as the older individuals that we will be studying, so that we don't introduce genetic variation that could influence our results. So we are currently recruiting younger Ashkenazi Jewish men and women to our study. So how can you get involved? Well, you have to meet a few criteria. You have to have 4 grandparents who are Ashkenazi Jewish. You have to be in good health yourself, and not to have taken any oral or intravenous steroids in the last 3 months. Inhaled steroids are okay because they don't really get absorbed into the body to any major extent. We will get your informed consent to participate in the study. We'll ask you to give a sample of blood and complete a questionnaire about the health of yourself and your family. And in return, we will give you information about the different types of cells that are circulating in your blood, and a $50 gift card for your participation, which we will greatly appreciate. So, this is going to be one of the biggest and most comprehensive studies of aging ever performed in humans. We hope it's going to give us some really interesting insights into age and its effects on cells and the potential role of stress hormones, and we would be very glad to have your participation. So please get in touch if you would like to participate in this study. Thank you.